आजकल भी तो आप आए थे ना नहीं हाँ 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 देखो कैसे यहाँ पे आके ना कुछ बोर हो जाओगे क्योंकि कुछ दिखेगा नहीं है ना तो क्या अपने फोन से देखोगे चलो हाँ 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 Good morning, everyone.
Good morning, sir.
Morning. Achha, today is is sort of a revision class on the three lectures that you had on enzymes. So I would like you to suggest to tell me if you have any problems in particular area. Can you say one by one? I will note it down and then I will go to those slides to explain that. Hello, anyone? Does anyone have any problem till about the enzyme classes that I have taken till now? Okay, so most of the comments that I am seeing are that you seem to have understood everything about enzymes, but still we have 1 hour to kill. So what I will do is I will briefly rush through the slides that I have taken or I've demonstrated before and explained to you. So basically what we have learnt is that lot of all our physiology and our this thing metabolism is controlled by a set of reactions. Now these reactions do not play much not only play an important role in metabolism, but even for all things that is essential for life. For example, say signal transduction, like a hormone coming and activating some receptors, and that that does some enzymatic action for the second messengers to be released, and so on and so forth. So each and every by aspect of our life is controlled by chemical reactions, and because of thermodynamic constraints, the rate of reactions may be very slow. Yeah, hello? So what we had learned that if these reactions were to take place spontaneously, then their rate of reaction would have been very slow, which will not be conducive for life. So all reactions have usually, just a minute, all reactions can be favored if they have a negative free energy change. That means the free energy of the initial reactants should be more than the free energy of the final product. So there is a decline in the free energy of the system. Now, so if the reactions can take place in this direction, then what prevents it from going the reaction taking place spontaneously? And that is because of all reactions, they form a temporary transition state analog which has a higher free energy than either the substrate or the product. So this higher energy prevents any spontaneous reaction that would have otherwise taken place from the substrate to the product. Now the role of enzyme is to, I think I've missed some slides. The role of enzymes is to bring down, by stabilizing the intermediate, the transition state intermediate, it helps to bring down the free energy of the intermediate or transition state intermediate it brings down the free energy of that. And hence what happens is that, so now the molecules of the substrate have a lesser transition state to lesser free energy to achieve, so as to become the transition state analog, and once it has reached this place, it can always slide down, it can always form the product. So normally in a system, in a system above 0 Kelvin, there is lot of all the molecules are in an energized state. That energy comes from two sources. One is the energy that is there within the bonds of the molecule, and the energy that is there because of the molecules moving around and bumping against each other. Now in a system at a particular temperature, there will be the energy distribution, the total free energy distribution of the molecules of the substrate in the system is a Poisson distribution, which is approximately equal to the normal distribution. So what you see in the normal distribution is that you have a sharp mean, and on either side, I think I have missed that particular slide in the revision class. Okay, so I will just explain. So what happens is, the number of molecules, so in a normal distribution you will see there is a tail end on either side. Some molecules will be having higher free energy than the mean of the system, and some molecules have lower free energy of the mean. But a molecule which has a lower free energy doesn't mean that it will always remain as a lower energy, because it can collide with other molecules, exchange some free energy, and can go to the high energy state. Now if you see the normal distribution in an uncatalyzed reaction, very few molecules will be at this free energy state, so that it can easily form the transition state analog. But if by stabilizing this molecule, the free energy of this molecule of the transition state analog is brought down by enzymatic actions or catalytic reactions, then what happens? More number of molecules are present in the system at this level of free energy, and hence more number of molecules are capable of forming product. So the enzymes do not actually alter the free energy state of the substrate or the product. It alters the free energy state of the intermediate, transition state intermediate, and hence it alters the rate of the reaction. That you have to be very clear about. Now how? So now see how basically how we can, now we will see how the thermodynamic lessons that we have learned is actually helping in enzyme substrate interaction. Now two substrates or two molecules come to each other, clash against each other to form our product. Now in a normal system, in an uncatalyzed system, the molecules have to strike at a particular place on the topography, so as to put some bonds on stress and others on strain, so that some bonds can be broken and new bonds can be formed. Now in a random collision, the striking of these two molecules, a collision of these two molecules does not lead to always lead to the formation of the reaction, because the collision may be happening at a place other than where the bonds have to be broken or formed, and hence the reaction and hence the molecules again fly off without forming any product or without any change. Now what happens is, now if in an enzyme, when the substrate collides with the enzyme, it has to collide in a particular direction or in a particular format, so that the appropriate bonds are put to stress. Now that is one way of how enzymes help in. So enzymes not only help the rate of a reaction by lowering down the free energy state of the intermediate, but what happens is it also helps to bind to the substrate. It helps to bind to the substrate in such a way that certain bonds are, the appropriate bonds are put under stress, and hence old bonds can be broken and new bonds can be formed, and the chemical reaction can take place. Now what we had learnt yesterday is that, the last class, is that there are two models that were proposed. The lock and key. Initially the lock and key model was proposed, where the shape of the active site of the enzyme, or the catalytic and substrate binding site of the enzyme, is complementary to the shape and size of the substrate. Any problem? Go ahead. So the substrate, so the enzyme, the topology of the enzyme at the active site is complementary to the substrate binding site, and the products are formed. But this was this was disproved later on, because it was shown because the free energy concept, that the intermediate free energy intermediate transition compound having a higher free energy concept could not be included into this type of model. So the next model that was proposed, and which stands till date, is the induced fit model, where the enzymes are binding, the substrate binding sites of the enzymes are not exactly complementary to the structure of the substrate, and hence when the substrate comes in contact with the enzyme, it has to for efficient binding or formation of this transition state analog, both the enzyme and the substrates are modified in their three-dimensional structure. And this modification, naturally, since they are forced to bind to each other, this modification leads to the formation of an intermediate transition state analog, which is the enzyme substrate complex, and that has a higher free energy than either the enzyme or the substrate or the product that will be formed. Now so when the substrate binds to the enzyme in the induced fit model, it changes and it forces the enzyme to undergo a conformational change so as to accommodate the substrate, and at the same time this conformational change in the enzyme puts stress on certain areas of certain parts of this substrate molecule, and it forms an transition state conformation which is has a higher free energy than these two combined. And once this type of constraint is put, then what happens? Some of the bonds in the substrate molecule are stressed or strained, and they lead to the breakdown of certain bonds and formation of new bonds, leading to the formation of products which can then escape the enzyme, and the enzymes regain back its normal three-dimensional structure. So other than thermodynamics and only some amount of molecules, in the absence of enzyme, what would have happened is that this substrate would have collided with each other, with another molecule of the substrate, and then if the energy of the collision was good enough, then some parts of the substrate would have some bonds in the substrate would have been appropriate bonds in the substrate would have been placed under stress, and some of it would have broken down and formed a product in the absence of the enzyme. Now, in the absence of the enzyme, if you look at the substrate molecule, it can strike in different orientations, out of which only a few orientations, striking at a few orientations only, will lead to the formation of a appropriate product. What enzyme does is that not only it lowers the free energy of the not only the free energy of the enzyme substrate complex is lowered, it also helps the substrate, molds the substrate in the appropriate conformation so that the product can be formed, which is not possible in the absence of the enzyme. The third thing is that in our biological reactions, most of the reactions are usually hydrophilic. That means they occur in an aqueous environment. So in the substrate molecule, most of the substrate molecules are usually polar. So in a polar molecule, there will be water molecules binding on its surface on the substrate, which is known as the hydration shell. Now, if two such substrate molecules with their own hydration shell collides with each other, what happens is that the collision occurs between the water molecules between these two substrate molecules, and hence the chance of that collision giving rise to deforming the substrate so as to give rise to the substrate products is less. But once it is bound to the enzyme, what happens is that the binding of the enzyme and the substrate molecule and the conformational changes binding of the enzyme with the substrate molecule leads to the removal of those water molecules that are forming the hydration shell. So that now the appropriate bonds are put on stress by the conformational change in the enzyme, and hence the reaction can go on. So this induced fit model was then before this model, first the induced fit model was just hypothesized, and it was not experimentally proven, could not at that point of time, because measurement capabilities of very short-lived transition state conformational was not available. But now we have various such mechanisms, various such ways to study the structure of the transition state conformation, and one such was one of the first thing that came to be experimentally proved was the binding of hexokinase with glucose, where the structures of these were separately identified, were separately deduced by X-ray crystallography, and then a solution containing glucose and a crystal containing hexokinase and glucose was also analyzed by X-ray crystallography, and it was found that the structure of the enzymes, hexokinase enzyme, without the presence of the substrate and in the presence of the substrate were different, and this helped to deduce that this helps to confirm the induced fit model. Now then, we had also studied some of the mechanisms of how an enzyme has specific how the enzyme becomes specific for a reaction, and that is because of the topography which will accommodate only substrates of a particular structure. For example, both this trypsin and chymotrypsin both are peptides, but trypsin will only can in the substrate binding site will only can accommodate the side chain of arginine, and chymotrypsin, which is because it has a positive charge and the binding site on the enzyme has a negative charge, and the shape of the structure of that binding site is complementary to the binding site of the side chain of the arginine amino acid. And in chymotrypsin you have a hydrophobic patch, and it is large, and which is appropriate for binding of phenylalanine. Now this only if phenylalanine binds to this place in this pocket there will be sufficient interaction between the enzyme and the substrate that the enzyme will be deformed, and consequently the substrate will also be deformed to form new bonds. Now for example, if you have just a methyl group like in serine, what happens is alanine. So what happens if it is a small methyl group which is also hydrophobic, which can also come and bind to this pocket, but it will not interact with the enzyme, and hence that methyl group will not appropriately change the structure of the enzyme so as to form so that the enzyme can also constrain the substrate molecule and help to break and create new bonds. So that is why the substrate molecule should be present in the substrate binding site and in the catalytic binding site. We had also learned through this example where we have shown that from the previous slide, the chymotrypsin molecule, where it binds here, binding to the hydrophobic pocket of the enzyme leads to the change in the enzyme molecules. So what happens in the enzyme molecule? So there was a previously hydrogen bonding between here and here. Now what happens? This bond breaks. This hydrogen bond becomes a permanent bond, and this conformational change stresses this part of the molecule of the substrate, and there is a cleavage. That leads to formation of a secondary bond, which leads to cleavage at this region, and we have one free peptide, followed by a sort of a reversal of this previous reaction, in which the water molecule helps to reform, to bring the enzyme molecule to its original structure, and in the process the other peptide is also, the N-terminal peptide is also released. Now we have also learnt about the different types of enzymes that are there. Now, as I told you, that enzymes are classified by certain numbers which are known as enzyme commission numbers. Now they occur in 4 sets of digits. The first digit basically broadly states what is the enzymatic activity. If it is transfer of hydrogen and oxygen atoms, or acceptance or donation of electrons, they are known as oxidoreductase. Because it is not that they have two separate enzymes, one oxidase, one reductase class, because all reaction, any oxidation reaction is always accompanied by a reduction, because if a substrate is getting oxidized, the another substrate is getting reduced. They go hand in hand. So that is why the first enzyme and the most important set of enzyme has been named as EC1, which is oxidoreductase. Then you have certain enzymes which are involved in the transferring of a group from one substrate to another. Like for example, transfer of say the phosphate group from ATP to say glucose. So they are transferases. Then you have breakages of bonds. Breakages of bonds can occur by two mechanisms. One is hydrolytic or one is non-hydrolytic. If a breakage of formation of two products is formed by the breakage of a bond in the presence of water, then it is called hydrolase enzymes. And if the breakage of the bonds occur not by themselves, without the involvement of water molecules, then they are known as lyases. And other important enzymatic functions are isomerization, where there is an intramolecular rearrangement without any group coming from outside or any cleavage of the molecule. Of course, during intermolecular rearrangement, cleavage from one of a part of the molecule will occur and the group will be transferred within the same molecule. So that's why it is different from isomerases, are different from transferases or lyases. And then you have ligases, which is the joining of two molecules, and usually they are high energy reactions and they require the breakdown, they require the involvement of ATP. The second number in the enzyme combination classification are the type of bonds it breaks. Hydrolases can be of different, for example, lyases can be of different types. It can break a carbon-carbon bond, that will be 4.1. Then similarly, if it's carbon-oxygen bond, it's 4.2, 4.3, and so on and so forth. The third is what is the product that is usually it denotes the type of product that it is forming. And the fourth number is the nature of the substrate. For example, here the product that is forming is amino acid in this tripeptide aminopeptidase, whereas the substrate is the tripeptide. So the third and fourth usually denote the product formed and the substrate required for the reaction. Okay, now let's see. Okay. So what we have seen, now we have studied till now, what we have devised is that enzymes directly interacting with the substrate. Now there are so many reactions that the body needs to take care of or to process, but it is sometimes not possible for the amino acid side chains alone to form complex bonds or donation of electrons or accepting electrons or protons for that matter. And so the enzyme gets a helping hand from certain other non-proteinaceous factors which are known as cofactor or coenzymes, which has certain side groups which help the enzyme to take part in the chemical reaction, and these side groups on the cofactor or in the coenzyme help in the functioning of the enzyme. They form transition state intermediates between the substrate and the enzyme to form product, and they are non-peptide in nature. They are non-amino acids. They are not amino acids, and hence they are known as prosthetic groups. And without its presence, when this prosthetic group interacts with the enzyme, the protein enzyme, then it actually forms the holoenzyme or the complete enzyme, so that it can carry out its enzymatic functions naturally and efficiently. Now the enzymatic the enzymatic actions that happen that we have studied till now are basically of covalent interaction, as we have seen, and also by just simple donation or acceptance of protons or electrons. So, and that is so without formation of any covalent bonds between the enzyme and the substrate, and they are known as acid base catalyst, where the side chains donate a proton or accept a proton or accept an electron, donate an electron, or if the side chain, if it is, or metal ions as cofactor can also help in the enzyme action by exchanging of protons or stabilizing intermediate structures or exchanging electrons. Now, as you will all know, there are so many, and now enzymes have particular affinity for each substrate. Now all the tissues in our body may not require the same amount of the enzymatic reaction as it is usually as required by other tissues. So different enzymes. So there are certain different types, subtypes of these enzymes exist in different tissues. For example, lactate dehydrogenase exists in has 5 different forms, as I had given you, I had explained to you last year, and we will also have a practical on this. It has 5 different types of isoenzymes. These are called isoenzymes. Now, why does the body have so many isoenzymes? Because the isoenzymes, they catalyze the same reaction, but the kinetics will be different. So in some isoenzymes the kinetics will be fast, in other isoenzymes the kinetic will be slower. So those tissues which require a fast acting enzyme will have a particular type of isoenzyme, and those tissues which require a not so fast acting enzyme will have another set of isoenzymes. Now these isoenzymes are isoforms of enzymes are also have a similar function, but they are structurally different. The isoenzymes, that all the protein chains that form the enzyme molecule, some of these protein chains will be different in the case of isoenzymes. That means there will be 2 or more gene loci for this enzyme, and 2 or more different proteins will be formed, and then they will associate with each other to form multimers, and then the sequences are different, primary sequences are different, so they are called isoenzymes. Now the same enzyme molecule, if it has the same backbone sequence, amino acid sequence, but post translationally if they are modified, then they are known as isoforms. But functionally they are similar. That means both this type of enzymes, they usually catalyze the same reaction, but at a different kinetics and with a different enzymatic activity and different substrate binding activity. Now I want to clarify one thing, that I want to clarify one thing that I had said, that any thermodynamic reactions, in any reaction where there is a positive, any reaction will take place only if there is a decrease in the free energy. For example, you have this reaction, glucose with ATP forming glucose 6-phosphate plus ADP. Now the free energy change of this whole reaction is negative. That means this has a lower free energy. The substrate molecule, the product molecules taken together, remember the word taken together, these taken together have a lower free energy than the substrate molecules, both of these two taken together. Again, remember the word taken together. So when so the glucose becomes glucose 6-phosphate, what happens? Glucose, there is a net change in free energy of there is a net change in free energy in the positive direction, remember, because glucose 6-phosphate has a higher free energy than glucose. Okay, but the changing of ATP to ADP is definitely a negative free energy change, because the ADP molecule has a lower free energy than ATP. So what we see here, that in this reaction, one molecule is gaining free energy and the other molecule is losing free energy. Okay, but the sum total change of this reaction, is actually negative. So that means in the presence of hexokinase, glucose plus ATP will always form glucose 6-phosphate and ADP, and not the reverse reaction will not take place, because for the reverse reaction there is an increase, very high increase of free energy in the system. So if you have glucose 6-phosphate and ADP, and how much amount of hexokinase you add to the system, you will never get glucose and ATP in that system, because there is a net change of free energy, and there is a positive free energy change of this reaction. But in our body we have reactions where glucose 6-phosphate is converted to glucose, but there you have to remember it is something a reversal of this reaction. Glucose 6-phosphate is a high energy molecule, as I told you before, than glucose, so it can easily, so this reversible reaction is easily done by another set of enzymes, and where ATP or ADP is not involved. That means ADP is not formed to ATP in this reaction. So only the 6-phosphate bond, high energy bond, is cleaved and you are left with glucose. So reactions that are thermodynamically impossible can occur in our body in a different system using different sets of enzymes and sometimes different co-substrates, so as to give rise to the appropriate product that we require. For example, you can form glucose to glucose 6-phosphate and also from glucose 6-phosphate to glucose in the body, but they are not catalyzed by the same enzyme, and the co-substrates are required are different, and the co-products that are formed are different. Which brings me to another thing that I have to mention, is that another reaction, that is glucose 6-phosphate to fructose 6-phosphate, that has a standard free energy change in the positive direction. That means this forward reaction, glucose 6-phosphate to fructose 6-phosphate, is not preferred, because there is a positive change in free energy, whereas the fructose 6-phosphate to glucose 6-phosphate reaction will be preferred by this isomerase enzyme, because the reverse reaction will have a negative free energy. But in our body we do have glucose 6-phosphate to fructose 6-phosphate conversion in glycolysis, and that is possible because the actual free energy, this is the standard free energy, that the actual free energy of the reaction is not only the standard free energy, but also this term added to it, that is R into T into log natural of the products upon reactants, which means that even though it has a fixed value under given conditions, we can alter, the body can alter to some extent. Remember the word to some extent. It cannot, for example, in the previous reaction, by this mechanism hexokinase cannot convert glucose 6-phosphate plus ADP to glucose and ATP, but in many cases like here, where the standard free energy change is smaller, then by altering the concentration of the product and the reactants, this delta G can be made negative, and the reaction can proceed in the direction that the body wants to. For example, here the standard free energy change here is positive, but if this fructose 6-phosphate is removed from the system by the next step in the enzyme, where it forms fructose 1,6-bisphosphate, then the product concentration will decrease. Or if there is more glucose 6-phosphate coming into the system by conversion of glucose to glucose 6-phosphate by hexokinase, then the concentration of the reactants will be decreased, and in both cases what will happen, this will form a fraction, and hence this term will become negative. So, and if it is sufficiently negative so as to make this positive standard free energy change ultimately giving rise to a negative free energy change, then the reaction will take place. Now you will argue, then why can't the hexokinase enzyme, this enzyme, the hexokinase that I was talking of, this reaction, why can't then it take place by if you decrease the concentration of ATP or glucose from the system, then why doesn't this reverse reaction take place? Because the net free energy change in this direction is so highly negative that how much amount of you increase this or this in this and this, or how much you decrease this and this, that RT log natural product upon reactants will not be sufficiently negative enough to make the standard highly positive standard free energy as negative, and that is why these type of reactions are so called irreversible reactions. Remember, all reactions are reversible, but certain reactions are so called irreversible with ports. That means they are for all practical purposes irreversible in our body, because they have such a high level of free energy change that the reverse reaction will not be possible even by playing with the concentration of the substrates and the product. Now I think I have covered most of the things that are required to know, that you should know for your in the first year. These mathematical these things are not required for you, but I had included in the lecture to make it slightly more interesting, but it is not mandatory for you to know. So now I think I have covered the enzymes till date that I have studied, that I have taught you. So if do you have any questions, you can ask me now, because this is my last class on enzymes. Do you have any questions?
Sir, what is the difference between isoenzymes and isoforms?
Okay, so isoenzymes are, you see, both are enzymes, different enzymes catalyzing the same reaction, maybe at a different rate, or has different binding affinity to the substrate. But the basic difference is that the primary sequence, the structure of the proteins, are different in isoenzymes. That means the amino acid sequence of these enzymes, if they are different, they are known as isoenzymes. But if the amino acid sequence are same of the two enzymes, the only difference is that it is post-translationally modified. Then they are known as isoforms. For example, we can say as an example, like say glycogen synthase. If it is post-translationally modified to become phosphorylated, then glycogen synthase will be inactive or less active. Okay, so glycogen synthase and phosphorylated glycogen synthase, they are isoforms, you can say, because they are post-translational modifications. There is no difference in the amino acid sequence of these enzymes, whereas in isoenzymes there is an amino acid sequence difference. Is it clear? Okay.
Yes, sir.
Any other questions? So I think I am not seeing any queries coming up. So with this we come to the end of this class. Okay, thank you. यहाँ पे आजकल there's no system of attendance ना attendance? How do you take attendance? हाँ. अच्छा. हाँ, 12 of us. So, okay, I don't know because my habit is I start shouting because I sit loud.
Hello. Hello. Sir, it's asking for password, sir.
Password.
Sir. I'll get the password, sir.
Where's the next one? Where should we go? Check the guy.
ये माइक है ना? So just introduce me.
Very, very good morning, students. Am I audible? Sorry for the slight delay. I hope you all were there for Parthas's lecture. Am I audible?
Yes, sir.
Okay. So I hope you all were there for Parthas's lecture. Okay, I am Dr. Seelan, SR Biochemistry. So it's very nice meeting you all. I hope you all are safe and happy. So today we'll have introduction on enzyme kinetics by Siddharth sir. So I hope all you have come. What is the strength? Okay. So I hope they will all join soon. We'll just wait for 2 minutes. So let's hope they all join us soon and we'll have the start of the session. Good morning, Mahesh. Yes. Yes, sir. Yes, sir, you can do it.
Good morning, everyone. One of you all will have to take on.
Good morning, sir.
Yeah, good morning. Okay.
Good morning, sir.
Yeah, good morning. All right. So my name is Siddharth, and the next couple of lectures I'll be taking. So the topic that I'll be covering is essentially probably the heart and soul of biochemistry. That's enzymes, reactions, and everything else. All right. So the important thing to understand is, despite all the technological advances over here, the most important thing in biochemistry, which we'd like to see even in your answer sheets and discussions, is lots and lots of arrows going from here to there, lots of reactants and so on and so forth. Okay, so that's, like I said, that's what biochemistry is really all about. Okay, so one of the components that we'll be dealing with in detail is enzymes. Okay, so this is a topic which I generally cover, and I cover it not just from the point of view from an experimental point of view. I also cover this topic from a computational and a mathematical point of view. All right, the reason is because I myself have worked very extensively on enzymes and kinetics, especially with mathematical modeling, computational programming, server development, and so on and so forth. Okay, so a lot of this lecture will be part of that kind of we'll cover that aspect also, computational biology and bioinformatics of enzyme kinetics. Okay, so what I'll just give you is a very brief outline of what we'll be covering. I won't start anything right now. I'll just tell you what we're going to do in the next 2 lectures. Okay, so the first thing that we're going to cover is basic definitions. Okay, now remember, this is a very, very important topic for you, especially when it comes to your exams and viva. All right, this is the core of biochemistry, and these are things that you need to know, absolutely need to know. Okay, it's not just important from the biochemistry point of view, but as you move on to your second years and third years, you're going to also see that it has immense clinical relevance. Okay, so we'll start off with what are basic definitions. In this basic definitions, we are going to cover generally the glossary that is associated with enzyme biology. For instance, what is the order of a reaction? Okay, what exactly are rate constants? What is the turnover number? What is the velocity of the reactions? These are very, very simple phrases, but these are the parameters on which a lot of drugs and other pharmacological agents are evaluated. Okay, so if you don't understand this, you're really not going to be able to prescribe a drug or so on and so forth. All right, so you need to understand some of these from that perspective. Okay, so that's what we'll cover in basic definitions. Okay, after that we are going to talk of what is called enzymes as biocatalysts. Okay, so in a way, what you'll have seen and heard of enzymes at the moment is that it's just an arbitrary molecule which sort of floats around. Okay, we are going to deal with enzymes as actually molecular entities. In other words, what really gives an enzyme the enzymatic activity. Okay, what causes an enzyme to be an enzyme. Okay, so in this we are not just going to cover, and many of you will probably already know this, that most enzymes are no doubt proteins, but there are many, many other enzymes also. Do you know other macromolecules that might be considered as enzymes? Very good. Yes, ribozymes, an extremely important component. What about something else? Anything else? Okay, so there's something. Okay, so there, in addition to ribozymes, there are 2 very important ones. One of them is more a laboratory concept, but it's still very much in the mainstream. Okay, so like you have RNazymes or ribozymes, you also have DNA based enzymes. A difficult concept to imagine, but they nonetheless exist. All right, and the third one is what are called abzymes. Any idea what abzymes are? Just take a guess. Go ahead. What are abzymes? No chance? You're taking guessing too far. No. Antibody based enzymes. Alright? So in a way, you can actually take an antibody and engineer enzymatic activity into an antibody. Okay, so those are what are called abzymes. Okay, so we won't talk about those, in detail, but we'll talk about the classical protein based enzymes. Okay, so what really contributes to those enzymes, that we'll describe in the lecture. Okay, the other topic that we'll talk about is how do we ascertain whether a molecule or rather a protein molecule will ever be an enzyme or not. Okay, somebody hands over the enzyme to you and say, look, it's an enzyme, you'll be happy. Correct? But what if you didn't know it was an enzyme? You were just given a protein molecule. One is, of course, you go to the lab and you sit and do a whole lot of experiments and see what it does. But is there any other way? Is there any other way of predicting whether or not a protein molecule will have enzymatic activity. Okay, so that is really the basis of an in silico assessment of protein sequences as enzymes. Okay, so what are these enzymes? How do you ascertain whether a particular molecule will be even an enzyme, right? Because these days there are so many molecules available, it's always good to have a head start. Okay, so of course you need experimental validation and so on and so forth, but it helps if you could narrow the space down a little. Okay, so that's where this aspect comes in which you can actually detect or shorten that kind of thing of ascertain whether an enzyme behaves like an enzyme or not. You can do this in silico. All right. Okay, that is so far insofar as about the enzymes. Then we are going to cover how do enzymes really behave. In other words, what is their kinetics? Okay, so kinetics really means how does it behave when it is in the cell, when you analyze it in vitro. So those aspects are what are called enzyme kinetics. Okay, and if you, like I said, I'm going step by step, because if you don't understand the glossary that is associated, for instance, rate constants, rate of the reaction, velocity, turnover, you're never really going to understand what kinetics is all about. Okay, so that's why this is very, very important. Okay, then in terms of kinetics, you also have different types of kinetics. Okay, so we are going to cover different types of kinetics over there. And in this case, when you're going to compare kinetics, again, this is a very, very important question for you. What are the different types of kinetics? How can you differentiate between them? So it's important that when you're differentiating two entities, you have to have a set of common points to actually differentiate one against the other, right? So therefore, these common points are, like I said, the parameters, which we would have talked about right in the beginning, right? So types of kinetics and what are the differences that are there. Then we are going to talk about what is called, okay, so then we are going to go a little deep into a very, very important part of enzyme kinetics, that is the derivations of two very, very important classes of kinetics. One is what is called the classical Michaelis-Menten kinetics, and the other is what is called allosteric kinetics. I won't go into the definitions over here, we'll deal with that later, but here we are going to tackle it especially from a mathematical point of view. In other words, when you see a reaction happening in vitro, how can you actually model that in, on a pen and paper and on the computer to actually figure out certain aspects of it, because you need to do these analysis sometimes beforehand before you actually commit to a particular treatment or so on and so forth. Okay, so we are going to have to deal with a few basic definitions. Okay, and the definitions which we'll be tackling is basically how do you derive the parameters for Michaelis-Menten and allosteric. Okay, then we come to a very interesting aspect of enzyme kinetics, that is what are the applications of this. Okay, this is insofar as it's the intellectual part, the research component is, but does it really have any use? So we are going to have an important component of applications. In other words, we are going to see, okay, how do we use this information to actually really do something. Okay, and the most important application based is what is called inhibitory kinetics. Okay, what are inhibitors? We are going to deal with. So if you know what enzymes are and how they're going to behave either in vitro or within the cell, you also are going to have to know what are inhibitors. In other words, same pharmacological agents which can either enhance or limit activity. And since of course we are talking inhibitors over here, we are basically talking about it being limiting. Okay, so inhibitory kinetics is a very, very important component. Okay, here is the basis for, so there are different types of kinetics and inhibitory kinetics which we'll be introducing to. We'll be giving you graphs to solve. Okay, we different types of things which are important from your practical point of view as well. Okay, and finally we are going to talk of regulation. Now enzyme regulation really can be considered twofold. One is what really regulates an enzyme's activity. Okay, so you have an enzyme, we know it is going to catalyze a reaction. What helps it or what does not help it? Okay, so what are the physical, the chemical, and sometimes the biological factors that either promote activity or in many ways inhibit that kind of activity? Okay, so this is about enzyme regulation. Then you have what are called activation factors. Okay, so activation factors will be, actually we are not going to talk of the enzymes in this case, we are going to talk of what are the factors which could again mean very, very small molecules, physical conditions that actually influence enzymatic activity. And this is a very important component, because when you're talking of activation factors, we're going to be dealing with the small molecular changes which can influence enzymatic activity. Okay, so much so for what we're going to cover in terms of this. Now I'm going to ask you a simple question. What do you think are some of the nice applications of understanding enzymes? Maybe you could think of something that is generally applicable, but I'll be really happy if I can hear something that is maybe a little esoteric, something that is out of the box also. Let's have your thoughts on the applications of enzymes.
Drugs.
Done. Accepted. Anything more? What else? Why do you need to know enzymes? Where else will you find enzymes?
So we're using biological catalysts in industries.
Okay, industrial applications, excellent. Many of the in fact biomass generators, or the biofuels, so to speak.
Clinical application.
Yes, clinical application. Then in so far as drugs go,
Genetic engineering.
You mean enzymes in genetic engineering or genetically engineered enzymes?
Sorry?
Yeah. So now one thing you'll have to understand in biochemistry is that you have to be very specific with your terms. Okay, so are you talking about genetically engineered enzymes or enzymes in genetic engineering?
Genetically engineered enzymes.
That is the same as recombinant enzymes. What about the other way around? Isn't that an important part?
Yeah, that is also important.
Can you name some? Can you name some enzymes in genetic engineering?
Restriction.
Yes. Just
ligases
exactly. So you,
and DNA. Sorry, proteases and lysosomes. Sorry.
yeah, absolutely. Very good.
Lysozymes.
Right. So you see, there's a difference between genetically engineered enzymes and enzymes used in genetic engineering. Right?
Yes.
So yes. Okay. Anything else? That's good. Let's have some more. Where else will you find enzymes or the uses of enzymes? Don't restrict yourselves to the simple samples and models. Think of something more. No?
The cleaning of nature.
Very good. Cleaning of stuff. Yes, that's an application of genetically engineered enzymes, where you can use and genetically engineer enzymes to clean out all spills and all that. Okay, granted. What else? More. Come on. Let's have something. You're moving out of the box, but you're still there at the border of the box. Come on, move out of the box. Anything?
Cleaning the environment like oil spills or something.
Done. I've given that. I've accepted that. More.
Clarifying fruit juices.
Which one?
For clarifying fruit juices.
Okay, well, that has a lot of industrial applications. What else? Come up with something more, no? Anything? Have you heard of Radiococcus radiodurans?
No, sir.
No? Not at all? Okay. What about astrobiology?
Yes, we used to send it to the outer space planets.
Fair enough. But that is outer space planets. Can you think of a way you can, before you move into outer space, you need to do some stuff beforehand here, no, on Earth?
Yeah, that would be done with the help of asteroids and all.
Asteroids? No. I'm saying on Earth, before going into outer space with these exotic enzymes, do you need to do something here on Earth?
Yes, sir. We need to make them radiation resistant toward
Fair enough.
cosmic radiations
Okay,
of
so
the
that's where the organism. What else? What else? Think. Can you mimic conditions out in outer space over here? Yes or no?
Yes, sir. Fairly enough input, sir.
Yeah, so you no, so I'm giving you a clue. Think astrobiology, think something else. What are we missing? Anything? Come on.
Sir, we could use enzymes to enable habitation in controlled conditions like high pressure or low temperature like that.
Granted, but how are you going to do that? How are you going to demonstrate that, at least in vivo or in vitro, whatever? That is in concept. How are you going to demonstrate that?
In vitro.
Yeah, that's true. But from where are you going to get these enzymes? Haven't you heard of
Prokaryotes, archaea,
Right, we are getting there.
archaea.
Yes. What?
This is thermosyncretic.
Very good.
Very
So,
thermophilic.
what are these organisms called?
So these are thermophilic organisms.
Granted, they're thermophilic. Some of them are halophilic.
Bacteria.
Halophilic.
Yes,
So, what is this category of enzymes called? These groups of organisms, they are called. I'll give you a clue. It starts with E.
extremophile.
Yes, very good.
Extremophile.
Yes, extremophile biology is such a wonderful application of enzymes in absolute, it's amazing. All these enzymes that are coming out in terms of, biotechnological applications have so many. Where have they come from? They've come from these extreme organisms. Okay, so extremophile metabolism is a very, very important component. Okay, so you see, enzymes are everywhere. Okay, great. I think that's about enough time that we have for. Do you have any questions on generally enzymes, or should we wait for our lecture on Thursday?
Sir, I read about some bacteria which is used for production of electricity.
Of course, yes. So you see, there are lots and lots of nice applications for it, right? Chalo, I think that's a good enough introduction. So we'll cover some of the slightly more mundane stuff on Thursday, but we'll talk about this set of organisms also a little in detail. All right? Okay, I think I'll sort of exit the meeting now. We'll see you on Thursday then, right?
Thank you, sir.
Yeah, okay. Thank you so much. Thank you.
Yes, sir.
Sure.
Thank you, sir. Yes, sir.
मैं जानू कितनी मेडिकल मैंने पता ना है